So welcome back to the course. Now we're gonna explain or see why and how polyploidization occur. So just a reminder on the plant structure first. You know that there is something important for plants, which is the apical meristem, which are stem cells, and they give rise to vegetative tissues like leaves, but also sexual organs like flowers that will then give the seed and the next progeny. It was actually thought that polyploidy was occurring in the apical meristem, so you will have chromosome doubling in this somatic tissue, and then of course this polyploidization will be transmitted to the sexual organs and then to the next generation, and so on and so forth. Nevertheless, some years after that, people started to realize that polyploidization occurs via sexual means and via what is called unreduced gametes. So that has to do with meiosis, and that's what is shown here. So just a reminder, meiosis is, let's divide it in two main phases, meiosis 1 and meiosis 2. In meiosis 1, the homologous chromosomes pair with each other, and this pairing is based on a sequence recognition. And this pairing will allow them later on, like you see here, the separation, like in spatial separation within the cell, and then the first cytokinesis will happen, leading to two first cells. And then during meiosis 2, this time what happens is that the sister chromatid from the same chromosome here separates, which in the end leads to 4 haploid cells if you start it as diploid. Okay, but sometimes things don't work that well. For example, let's take here a germ cell, so diploid 2x with 2 different chromosomes, which makes 4 chromosomes total. Meiosis normally should happen, leads to n equal x equal 2, so 4 haploid cells. But sometimes meiosis doesn't work well, and then either through genetic factor like hybridization, we'll talk about it later, also environmental factor like temperature influence meiosis. And what does that mean that meiosis doesn't work? Basically, meiosis doesn't lead to the division of the number of chromosomes, and you end up in the gametes with the same number of chromosomes as you had in a germ cell. And of course, if those two gametes fuse together to give a new zygote, a new progeny, then this progeny will have double the amount of chromosomes compared to the germ cell of the parents. And that leads to polyploidy. This naturally occurs, but at extremely low frequency, up to 0.4% in Brassicaceae or 0.9 in other species, but it remains pretty low. And then two main pathways can be affected in the meiosis. So here again, normal meiosis. So you have production of the sister chromatids, separation of the homologous chromosomes in meiosis 1, and then separation of sister chromatid in meiosis 2, leading to 4 haploid cells. Okay. The first problem that can happen will affect meiosis 1 actually, which means that the homologous chromosomes won't separate spatially. They will stay in the same cell, which of course means that during meiosis 2, you will have still separation of sister chromatids in these two cells, but then the two chromosomes will be in the same cell, leading to the double amount of chromosomes. And when you talk about second division restitution, then it's all the way around. Meiosis 1 works well, so you have separation of the homologous chromosomes, but then the sister chromatids still separate, but don't separate spatially, which means they will remain in the same cell, leading to the same chromosome with the two sister chromatids separated, but it leads in the end to the same, which is unreduced gametes. Okay. And then another point is you have two different kinds. You can have autopolyploidy or allopolyploidy. Autopolyploidy means that you're going to have this polyploidization event occurring within the same species. While in the case of alloploidy, you probably will have hybridization first and then polyploidization. So it means allopolyploidy is coming from two different species, at least. Then, what people think happens in terms of when we look at autopolyploidy, sorry. So it could be one theory that has been proposed is basically, let's say you have a diploid parent species, meiosis fail, will lead in the end to diploid gametes from both the paternal and maternal side, let's say, which leads to, of course, a zygote, which becomes tetraploid because double here means double there. Okay. There is a problem with that. I told you that the rate of this happening naturally is maybe 0.5% in average. For the probability that two unreduced gametes happen under these conditions, this one and this one, is then 0.5% to square, which means this probability, which makes it quite low frequency. Of course, as I told you, some environmental factor like temperature can increase that, but let's say that this is quite an unlikely event to happen. Another suggestion could be that, okay, you have a parental species again, but this time you have meiosis failure in only one of the gametes, which is higher, which is then 0.5% probability. Still low, but much higher than this one. And then the other one, let's say the female here, is not unreduced, so it's haploid as normal. And then these two will meet and create a triploid offspring. Then what can happen if it's self-cross or back-cross to the parent is that after some time you get a tetraploid offspring. I will come back to this after and why this can happen, but that's the two theory proposed. Now let's look at allopolyploidy. This is a bit more tricky. So you have here the history of wheat that has been reconstituted now quite well, and what we know is that basically you had these two ancestral species, so Tridicum monococcum with potentially Tridicum crc or maybe Aegilops peptoides. So those two species were diploids, and let's call that this one had the genome A, so AA, and BB for the other species, because they are both diploids. Both have the same chromosome number of 7, so diploid total 14, okay? What they thought is at some point they hybridized, leading to this AB hybrid with the same chromosome number, okay? And then at some point there were polyploidization events, like what you have here and what you can see here. And this is actually durum wheat, so the wheat, so the wheat that is used for pasta, for example. And this is tetraploid AABB. Then there was another hybridization event with this time Tridicum taoshi, genome, let's call it D in this case. So this one is diploid, and then hybridization between a tetraploid and a diploid leads to a triploid, okay? So ABD. And then again, another polyploidization event happened, leading to the bread wheat, so the one used for the bread, leading to these hexaploids and having the genome AA, BB, and DD. Okay, that's quite striking because it's a very short time in the end. In terms of evolution, it's a very short time of things happening. We're talking about 10,000 years or maybe 15,000 years. How come polyploidization can be that frequent in this case? Well, actually, it's quite interesting. So let's talk about, again, if I come back to here, let's talk about this hybrid here that happened 10,000 years ago. This is actually not a real diploid, in a way that it's made of two different genomes, A and B, and these two genomes have chromosomes that are called homeologous, not homologous. What homeologous means is that they have a shared ancestry, so which means that at some point they were the same chromosome, but then when the species split, they evolved separately. And then they evolved so far that they were in two different species, and then the genetic distance between them increased a lot. So if you look at what happens in this kind of hybrids during meiosis, so S phase again, so production of sister chromatid, okay, the same. Something important is during meiosis 1, so during the chromosome pairing, I told you that the chromosome pairing is sequence based. So two chromosomes of the same sequence or very similar sequence will pair very easily. But as I told you as well, homologous chromosomes are quite distant in terms of genetics. And that means that basically the sequence-based pairing cannot happen. So that's what you have represented here. So if you don't have pairing, you don't have separation of the chromosomes, which basically means that you will end to unreduced gametes. And that actually happens to quite a high frequency. And for example, in these papers, they show that in brassica, this happened up to 34% of gametes are unreduced. So 34% of hybrids gametes are unreduced. And of course, now if you think both female and male needs that to produce a polyploid or tetraploid, then it gives you a probability of 12%, which is pretty high compared to what we discussed about autopolyploidy. And that actually led to propose that alloploidy is actually the main road to polyploidy in general, that you have much more frequently allopolyploids than you will have of autopolyploids. Yeah, just a summary. If you have, of course, two unreduced gametes of this AB hybrid from the weed, then you get to this tetraploid when these gametes fuse together. And this was the durum wheat then. So, time to summarize. So, I will ask you on this quiz to tell me what does it mean to have, or what are unreduced gametes, and then how to write them using the formula, using the numbers that we discussed already, so X, N, or whatever. Then what is the difference between autopolyploid and allopolyploids? The difference between homologous and homeologous chromosomes. And then a question a bit tricky. So we discussed about allopolyploidization, how it happens and how it leads to unreduced gametes. My question here is, when unreduced gametes happen in hybrids, does it lead to a first division restitution or to a second division restitution? And finally, something we didn't talk about it yet, but there is a term called aneuploidy, and you can write it that way, 2n plus 1. Can you tell me, can you evaluate what does that mean in general terms? Good luck.